So let's review now what we can do to manipulate this tumor microenvironment with therapeutic ultrasound. We start again. Context, tumor microenvironment, this complex interplay between immune cells from the adaptive and innate immune system, the cancer-associated fibroblasts, the extracellular matrix, the cancer cell, the blood vessels. And what can we do? Potentially, we can act at very different levels. We can modulate the danger signal that will be released. We can disrupt the stroma. We can destruct directly the cancer cells that will result in more release of cancer cell antigens. We can modulate the cytokine expression, and at the level of the blood vessel, we can increase the permeability for drug delivery. We can modulate the adhesion molecule, the cytokine, the chemokines to enhance the trafficking of the immune cells into the tumors. So let's review some examples. This first one is thermal ablation. This is a melanoma model treated with a sparse thermal ablation strategy. And what was observed is that you could have not only a tumor growth control of the treated tumor, but also a partial distant tumor growth control. And this is very important because it suggests that you can treat at one site, have an effect at a distant site, so you're able to generate a response that is not only local, but is also systemic. Another very interesting observation is that in the case of thermal ablation, it seems that what is happening is not necessarily into the core of this necrotic ablated area, but more on the periphery, or the periablative zone. In this example, an increase of infiltration by dendritic cells. Example number 2, mechanical ablation, histotripsy. This is a rat model of liver tumors. The tumors were partially ablated, 50–75%. But it resulted in a complete tumor regression in more than 80% of these rats. Again, meaning that even if you treat locally, the response you are triggering seems to be more systemic. We can now use ultrasound to deliver drugs, and this is an example of delivery into the brain to treat brain tumors. So this is blood-brain barrier opening. No sonications, one sonication, two sonication to deliver anti-PD-1 antibody that were tagged, and you can see how multiple sonication can improve the accumulation of the antibody in the brain, and that resulted in an improved survival of these treated animals. We can also do it to deliver cellular therapy. So this to deliver, in this example, CAR T-cell. Again, T-cells that have been engineered to express this chimeric antigen receptor. No sonication, sonication, and you can see accumulation of this CAR T-cell in the brain. That also resulted in a therapeutic efficacy with an improved survival of these treated animals. Now, let's talk about combinations. So combining therapeutic ultrasound with immunotherapeutics. The idea being that sometimes in some models, these preclinical studies, right, the tumors are not responsive or only partially responsive to the immunotherapeutics that you're going to use. And the question is, if we add a layer on top of that, an ultrasound therapeutic ultrasound intervention, can we improve the response to these immunotherapeutics? In this example, this is a triple-negative breast cancer model, which has a very immunosuppressive tumor microenvironment that are treated with gemcitabine, which is a chemotherapy that will target more specifically some components of this immunosuppressive TME. And what we see that this is thermal ablation, sparse thermal ablation. We see that the response to gemcitabine is improved compared to control, but more significantly improved when it is combined with a therapeutic ultrasound intervention. And here I would like to pause just 1 second to talk about a very important experiment that need to be performed in kind of studies, which is to try to disentangle the effect of the ablation, debulking, against the effect of really triggering an immune response. And to do that, what you can do is to deplete specific parts of your immune system. For example, you can deplete the CD4 positive T cell or the CD8 positive T cells, or as in this example, you can use animal, these rag-1 mice, that lack mature B cell and T cells. And what you see, this is the same model, the same combination. Now, if you compare the animal that received only gemcitabine to the animal that received the combination of gemcitabine and therapeutic ultrasound, you lost the advantage of the combination. So it's not a causal demonstration of or mechanistic understanding of what exactly is going on, but it's a proof that the immune response is at least partially involved in this combination, in the response to this combination. And these are really important experiments to perform. Another example, histotripsy combined with immune checkpoint inhibitors. This example is again a two tumor model. Only one will be treated with histotripsy. These are neuroblastoma inoculated subcutaneously that are left to grow very large so that they become refractory and resistant to non-respondent to this combination of anti-CTLA-4 and anti-PD-L1 antibodies. And you can see that animals don't respond or really minimally respond to this combinatorial checkpoint inhibitor therapy. Now you add a layer of histotripsy on only one of the tumor, and you see how this animal will respond. First, the both tumor will regress, and in 70% of the animal disappear, leading to this survival curve that could be followed up to almost a year. So very striking data that suggests, or that demonstrate, I would say, first that you can induce a systemic immune response, and this response is very durable. So the idea of these combinations is also to really derive a process of auto-vaccination. And again, if we go back to really one of our initial questions, how to teach the immune system to recognize the tumors. And one design that is proposed is to do it in a multi-step approach. To start with priming the immune system, for example, with CpG, so to prepare the immune system in some sort to be ready when the injury will arrive. Step 2 then will be, for example, thermal ablation or partial thermal ablation. And because you prime the immune system, the cross-presentation and the activation of the T cells by the dendritic cells will be highly effective. And then, so if we follow our immunity cycle, T cell migrate and reach the tumor and start attacking the cancer cells. And this is where we can have a third layer of therapy, which can be, for example, immune checkpoint inhibition with anti-PD-1, anti-CTLA-4, to really unleash the potential of these activated T cells. And finally, the last part is to discuss sonogenetics. Sonogenetics, it's a combination of synthetic biology and ultrasound to develop biomolecular tools to allow ultrasound to connect directly to cellular functions such as gene expression. One way to approach this, to understand this, is thinking about heat shock promoters. We know that they respond to mild hyperthermia, so mild hyperthermia 42, 43 degrees, activate the promoter that will in turn trigger the expression of the heat shock protein gene. Now, if you can construct a circuit that will be under the control of a heat shock promoter and you apply hyperthermia with ultrasound, well, you have a way to only locally trigger the expression of your transgene. And this has been applied to engineer probiotics that can locally deliver tumor therapy. So these bacteria were engineered to release immunotherapy only under hyperthermia condition, in that case to release anti-CTLA-4. We know that these bacteria are tumor homing, so they will accumulate in the tumors, and then all you have to do is to induce hyperthermia with ultrasound, and you can see that this will result in a very significantly improved tumor growth control. Second possibility is to use this approach to control CAR T cell activity. So these CAR T cells have been engineered to express their CAR, their chimeric antigen receptor, only under hyperthermia conditions. And so looking here, you see the expression of the CAR is triggered when you induced hyperthermia, for example, with therapeutic ultrasound. This is a transient mechanism that will return to baseline after 24 hours and that you can repeat. And in terms of therapeutic efficacy, in this study, they used the two tumor models. Only one was treated with the combination of CAR T and hyperthermia induced by therapeutic ultrasound, leading to drastic tumor growth control in the treated tumor, but not in the untreated tumor. And this can have very important implication to limit the off-target toxicity of these CAR T cells, to limit because this CAR and these T cells will be activated only at the site where you induce hyperthermia with ultrasound. So to summarize, we can use therapeutic ultrasound to improve the delivery of immunotherapeutics in brain and in solid tumor. This is effective and that leads to increased survivals. In terms of immune response, now that is induced by therapeutic ultrasound alone, following thermal ablation, and by that we mean thermal ablation of the whole tumor, it seems that it might be minimal, but more studies are needed. Mechanical ablation, sparse thermal ablation, non-ablative ultrasound modalities are potentially more promising. The combination of therapeutic ultrasound and immunotherapy, again, there is a lot of very encouraging data in preclinical models. Sonogenetics, spatial and temporal local delivery of tumor therapy. You can control the CAR T cell activity, and that could lead to the design of more safe and effective therapies. And really to conclude, we have all these elements of evidence that we can modulate the tumor microenvironment, that we can trigger, improve the response to immunotherapy, that we can trigger a systemic immune response and observe abscopal effect. We are still lacking a very clear mechanistic global understanding of what type of therapeutic intervention will lead to what type of immune response. And this is where preclinical studies and more biological understanding is still necessary.